On a 15 milliliter tube, write your name on a label and stick it on. In this case, since we don't have name tags, we are using stickers to indicate whose tube it is.
Okay.
Add 2.5 milliliters of sports drink in each cup. Drink the sports drink from a cup and do not swallow. Swish it around in your mouth for 1 full minute and gently scrape the insides of your cheeks with your teeth to release the cheek cells. Spit the sports drink with the collected cheek cells back into the cup and pour it into your labeled 50 milliliter tube. Using a plastic pipette, add 2 milliliters of cell lysis solution to your collected cheek cells.
Open so that it's
Cap your 15 milliliter tube tightly and turn it upside down 5 times. This will mix the cell lysis solution with the collected cheek cells. Allow the tube to stand for 2 minutes.
so we just mix our
At an angle, use the plastic pipette to carefully add cold 70% ethanol, running it down the inside of the tube. Add the ethanol until the total volume reaches 12 to 13 milliliters. You should have 2 distinct layers. Do not mix the cheek cell lysate layer with the ethanol layer.
My giraffe. Hi, giraffe.
Using your plastic pipette, transfer your precipitated DNA out of the 50 milliliter tube and into the pendant tube. Begin pouring the DNA from the end of the most excited strand in the ethanol layer. As you pour, the DNA will be drawn together. Do not move your pipette down into the cell lysate layer.
Mine does not have DNA at all.
If some of the DNA remains attached to the cell lysate layer, draw your pipette up until the DNA in your pipette detaches from that in the cell lysate layer. You do not need to transfer all of the precipitated DNA sample into your pendant tube. Before you release your DNA into the pendant tube, allow it to sink to the tip of the pipette so that it will enter the pendant tube. If the DNA does not sink, release ethanol into the 50 milliliter tube until the DNA is in the pipette tip. Release the DNA into the pendant tube and fill the remaining space with ethanol. Do not overfill. Okay.
We need a new pipette. Excuse you. My DNA is alive. The giraffe. Top. There's nothing there. DNA.
In conclusion, long strands of double helical DNA can fit into the nucleus of a single cell because DNA folds around proteins and compacts itself.
A sports drink is used to collect the cheek cells inside the water because sports drinks contain salt and are saline solutions compatible with the osmotic environment of the cell.
Cell lysis solution dissolves the phospholipid bilayer of cell membranes by forming water-soluble complexes with them, allowing a soup of cell contents called cell lysate to flow out of the cell. Thank you.